In this video, we'll discuss how to use various tools in Linux to align your data to a reference genome. In addition, by the end of this video, you will have essential information about assessing FASTQ file quality and steps you can take to improve the quality of your alignment. Hello, and welcome to another video on NGS data analysis. In our last episode, we briefly discussed 5 genomic file formats: FASTA, FASTQ, SAM, BAM, and VCF. These formats are all integral to the NGS data analysis. In this video, however, we will focus on FASTQ and FASTA files, as they represent the initial sequencing data we will work with. In general, during NGS, when you provide a DNA sample to the sequencing machine, it generates reads of the DNA fragment present in the sample. Each of these reads includes quality scores for each nucleotide, which are essential for downstream analysis. However, like any other laboratory equipment, sequencing machines can make errors. Technical artifacts, DNA sample contamination, and residual adapter contamination can lead to false positive or false negative results. Therefore, before proceeding any further, it is crucial to perform a quality control step on the FASTQ files received from the sequencing machine. One of the most popular tools for FASTQ quality control is FastQC. It generates comprehensive report, highlighting metrics like per-base sequence quality, GC content, sequence duplication, and adapter contamination. Here are key things to watch for. Per-base quality scores. Ensure most scores are at least above 20, ideally above 30. Sequence length distribution. Confirm consistent read length unless using variable length protocols. Adapter content. High levels suggest incomplete adapter trimming, which can interfere with alignment. GC content. Look for unusual deviations from expected GC content. It might indicate contamination or biases. Detailed information on each of the metrics from FastQC tool is present on their official website. When you encounter reduced pair-based quality scores, especially at the extreme ends of the reads, or when there is higher adapter contamination, you can use tools like Trimomatic to address these issues. Trimomatic is a user-friendly tool designed specifically for trimming Illumina sequencing data. It removes low-quality bases, adapters, and other unwanted sequences, ensuring your reads are clean and ready for alignment. Let's look at its basic syntax. A typical Trimomatic command looks like this. Here's what each parameter means. The PE means that the FASTQ files are paired-end sequencing data. For single-end data, you can use SE. You then need to specify the input FASTQ file. Since we have paired-end sequencing data, there will be 2 FASTQ files as input. For the output, you can specify 2 files for each input. One for the trimmed FASTQ file, which contains the reads that passed quality control, and another to collect the reads with low quality. The second file can be given an untrimmed extension for the name. The Illumina clip option. This utility removes adapter sequences, and it requires an adapter file which contains adapter sequences, mismatch thresholds, and thresholds for paired-end alignment scores. And finally, you need to specify the sliding window option. This tool trims reads dynamically, cutting once the average quality in a window falls below the specified threshold. Therefore, by running Trimomatic, you can trim low-quality ends and remove adapters, significantly improving the quality of your FASTQ files. Now that we've cleaned our raw sequencing data, it's time to align those reads to a reference genome. But what exactly is NGS data alignment and why is it crucial? Alignment is the process of taking millions or even billions of short reads generated by the sequencer and mapping them to a reference genome. This step allows us to identify where each read originated and is fundamental for downstream processes like variant calling. But alignment isn't always straightforward. Sequencing errors, genomic variations like SNPs and indels, or repetitive regions in the genome can make mapping challenging. That's when advanced algorithms come in. Alignment tools rely on specialized algorithms to efficiently map millions of reads to a reference genome. Most tools use one of the following two approaches. The seed and extend approach. Tools like BWA and Bowtie first identify short seed sequences in the read that match the reference, then extend the alignments while allowing mismatches and gaps. The suffix array or Buris-Wheeler transform or BWT approach. These methods compress the reference genome, enabling rapid search and alignment while minimizing memory usage. BWA and Bowtie both use BWT for short reads. For long reads, which are more error-prone, tools like Minimap2 use graph-based algorithms to tolerate higher error rates while still achieving fast and accurate mapping. To perform alignment efficiently, tools require the reference genome to be pre-processed into an index format. An index file acts like a roadmap, allowing the aligner to quickly allocate regions of the genome for comparison. For example, when using BWA, you must first create an index for the reference genome. But keep in mind that you have to perform this step only once for each genome assembly. This process generates several auxiliary files to help the aligner rapidly navigate the genome. Without an index, the aligner would need to scan the entire genome for each read, a process that would be computationally expensive and time-consuming. After preparing the index, aligning the FASTQ files should be straightforward. To perform alignment, you only need to specify the reference genome FASTA file and the FASTQ files. In the BWA syntax, we have also included a few additional options, like the MEM option. This is the BWA algorithm designed for short-read data alignment. The -M option marks shorter split hits as secondary alignments. This option is useful when a read can align to multiple locations in the genome. BWA identifies the primary alignment or the best alignment and marks the other alignments as secondary. This information is critical for downstream data analysis. The -T option specifies the number of CPU threads to allow BWA to use. More threads lead to faster alignment. Another option you can also include is -R, which allows you to specify read group information to be embedded in the SAM file. This step is important for the downstream analysis process, so it makes sense to add it at this stage. The file resulting from this step is a SAM file, which is in plain text format. Once alignment is complete, you'll need to evaluate the results. Key metrics include coverage, which is the number of reads mapping to a particular region, and the mapping quality, which is the confidence score for each alignment. High-quality alignments ensures better downstream analysis. One of the most popular tools that can be used for the quality control of aligned reads is called SAMTools. The syntax for this purpose looks like this, and it basically removes the reads with mapping qualities lower than 20. The output of this command is a BAM file. One cool trick that you can use here is the pipe function. Represented by the vertical bar, the pipe function is a powerful feature in Linux. It allows the output of one command to be sent directly as input to another, creating an efficient, streamlined workflow without intermediate files. When processing ingest data, you can use pipes to align reads with BWA and directly remove the reads with lower mapping quality of the resulting alignment with SAMTools, saving both time and storage. So that's it for today. We've covered how to efficiently align your sequencing data using BWA, clean and process the data with SAMTools, and leverage the power of Linux pipes to streamline your workflow. Mastering these tools is essential for any NGS pipeline, and with practice, you'll be able to handle even most complex datasets. If you found this video helpful, don't forget to like, subscribe, and leave a comment below with your questions or suggestions for future uploads. And like always, keep exploring the wonders of science and take care.